In the vast ecosystem of the human body, trillions of microorganisms silently orchestrate a symphony of functions critical to our health. These microscopic inhabitants, collectively known as the microbiota, wield a profound influence on our physiological processes, immunity, and even our susceptibility to diseases. As research delves deeper into the intricate relationship between microbiota and human health, it becomes increasingly evident that this microbial community plays a pivotal role in the onset, progression, and treatment of various diseases. The microbiota unveiled. The microbiota, predominantly residing in our gastrointestinal tract, comprises an assortment of bacteria, fungi, viruses, and other microorganisms. This diverse ecosystem co-evolves with its human host, establishing a delicate balance crucial for maintaining homeostasis. However, factors such as diet, lifestyle, antibiotic use, and environmental exposures can disrupt this equilibrium, leading to dysbiosis, a state characterized by alterations in the composition and function of the microbiota. Microbiota and disease dynamics. The intricate interplay between the microbiota and human health is exemplified by its involvement in a spectrum of diseases. Inflammatory bowel disease, IBD. Dysbiosis of the gut microbiota has been strongly implicated in the pathogenesis of IBD, encompassing Crohn's disease and ulcerative colitis. Imbalances in microbial diversity, accompanied by an overabundance of pro-inflammatory species, contribute to chronic intestinal inflammation, exacerbating disease progression. Metabolic disorders. Emerging evidence underscores the role of the gut microbiota in metabolic diseases, such as obesity and type 2 diabetes. Dysbiotic alterations can disrupt energy metabolism, promote inflammation, and influence host signaling pathways involved in glucose and lipid homeostasis, thereby predisposing individuals to metabolic dysfunction. Neurological disorders. The gut-brain axis, a bidirectional communication network between the gut microbiota and the central nervous system, has garnered significant attention in neurology. Dysregulation of this axis has been implicated in various neurological conditions, including depression, anxiety, autism spectrum disorders, and Parkinson's disease. Perturbations in microbial composition and metabolite production can modulate neuroinflammation, neurotransmitter synthesis, and behavioral outcomes, thereby influencing disease susceptibility and progression. Autoimmune diseases. Autoimmune disorders such as rheumatoid arthritis, multiple sclerosis, and systemic lupus erythematosus are characterized by dysregulated immune responses targeting self-antigens. The gut microbiota exerts a profound influence on immune development, tolerance, and regulation. Dysbiosis can trigger aberrant immune activation, disrupt mucosal barrier integrity, and promote the release of inflammatory mediators, thereby contributing to autoimmune pathogenesis. Cancer. Mounting evidence suggests a link between the gut microbiota and cancer development, particularly colorectal cancer. Dysbiosis can foster a pro-inflammatory milieu, induce genotoxicity, and modulate host immune surveillance mechanisms, thereby promoting tumor initiation, progression, and metastasis. Harnessing microbiota for therapeutic interventions. The pivotal role of the microbiota in human disease has spurred intense interest in leveraging these microbial communities for therapeutic interventions. Strategies such as probiotics, prebiotics, postbiotics, fecal microbiota transplantation, FMT, and microbial-based therapeutics hold promise in restoring microbial balance, modulating immune responses, and ameliorating disease pathology. Looking ahead, unraveling the microbial mysteries. As our understanding of the microbiota continues to evolve, so too does our appreciation of its multifaceted implications for human health and disease. Integrating microbiome-based approaches into clinical practice holds tremendous potential for personalized medicine, diagnostic biomarkers, and novel therapeutic avenues. However, unraveling the complexities of the microbiota-host interaction necessitates interdisciplinary collaboration, innovative technologies, and large-scale longitudinal studies. The microbiota represents a dynamic and indispensable component of human biology, intricately intertwined with our health and susceptibility to disease. Challenges and future directions. Despite the remarkable progress in microbiota research, several challenges and unanswered questions persist on the road to deciphering the complexities of the microbiota disease nexus. Microbial diversity and dynamics. The vast diversity of microbial species and their dynamic interactions within the human microbiota pose significant challenges in characterizing and understanding their roles in health and disease. Longitudinal studies incorporating multi-omics approaches are needed to elucidate the temporal dynamics of microbial communities and their functional contributions to disease states. Causality and correlation. Distinguishing between causal relationships and mere correlations in microbiota disease associations remains a major hurdle. While observational studies have identified microbial signatures associated with various diseases, mechanistic insights into how specific microbial taxa or metabolites influence disease pathology are often lacking. Experimental models, including germ-free animals and in vitro co-culture systems, are indispensable for elucidating causal relationships and underlying mechanisms. Host microbiota interactions. Deciphering the intricate interplay between host genetics, environmental factors, and the microbiota in disease susceptibility and progression is a daunting task. Integrative approaches combining genetics, microbiology, immunology, and environmental sciences are essential for unraveling the complex web of host microbiota interactions and their implications for human health. Therapeutic translation. While microbiota-based interventions hold immense therapeutic potential, several challenges must be overcome to realize their clinical utility. Standardization of intervention protocols, optimization of microbial formulations, identification of responder phenotypes, and long-term safety and efficacy assessments are paramount for the successful translation of microbiota-targeted therapies into clinical practice. Ethical and regulatory considerations. The ethical implications of manipulating the human microbiota, particularly through interventions such as fecal microbiota transplantation, FMT, and microbial-based therapeutics, warrant careful consideration. Regulatory frameworks governing the development, testing, and commercialization of microbiota-targeted interventions must balance innovation with patient safety and ethical concerns. Pioneering the microbial frontier. In the quest to unravel the mysteries of the microbiota and its impact on human health and disease, we stand at the forefront of a new era in biomedical research. By embracing the complexity of the microbiota-host interface, harnessing cutting-edge technologies, and fostering interdisciplinary collaborations, we can unlock the full therapeutic potential of the microbiota for addressing a myriad of human diseases. As we navigate the uncharted terrain of the microbial frontier, let us remain steadfast in our commitment to advancing scientific knowledge, translating discoveries into innovative therapies, and ultimately improving the lives of individuals affected by microbial-related diseases. In the symbiotic relationship between humans and their microbial counterparts lies a wealth of untapped potential, a potential that holds the key to unlocking new frontiers in medicine and realizing the promise of precision health for generations to come.